And we are moving on to session 1, which is Innovative Technologies and Interventions. This will be chaired by Dr. Jihan and Dr. Ghislaine from Monash. To all our first session speakers, if we have any technical issues, please raise them immediately with any member of the organizing committee, and we'll sort this out. We are keeping time, and you'll be asked to finish on the dot at the 10-minute mark. Thank you.
Oh, hi, everyone. My name is Jihang. I will be chairing this session together with Glenn. We have a great line of speakers. The first speaker is Christine Kim, and she's going to talk about development of hypoxia-activated prodrugs of second-generation analogs of beta-quinine for treatment of latent tuberculosis. If you could share the screen, that would be great.
Is that sharing everyone?
Yes, I can see your screen.
Oh, okay,
could you put it in full screen mode? Yeah, awesome. All yours.
Is that visible for everyone?
Yes, looks good.
Okay, yes, then I will make a start. Morning everyone, my name is Christine and I'm a fourth year PhD student at the University of Auckland. So the name of my project and the talk this morning is the development of hypoxia-activated prodrugs of second generation analogs of bedoxolone for treatment of latent TB as introduced. So tuberculosis is a chronic disease caused by Mycobacterium tuberculosis, and it most commonly affects the lungs. It is a major health problem in developing countries, and currently it is estimated that a quarter of the world's population, or 2 million, is infected with MTB. Last year, it was reported that 10.8 million were affected by active TB disease, and of these, 1.25 million were fatalities. So 5 to 10% of carriers of MTB will become sick in their lifetime, and multi-drug resistance, or MDR, is on the rise for TB patients. In human TB, granuloma is observed in both and latent forms of the disease. So it is formed when the MTB is contained by the adaptive. Oh, yeah, there we go. It is formed when the MTB is contained by the adaptive immune system, and it is characterized by a gradual depletion of oxygen. So dormant bacilli reside in a low oxygen or hypoxic environment of the TB granuloma, and the granuloma acts to contain the infection. Unfortunately, some bacilli can survive for many years in a dormant state. So granulomas are problematic as they hinder antitubercular drug penetration, resulting in accumulation of the antitubercular agent into the surrounding cells and increasing toxicity. Bedoxolone was approved by the FDA in 2012 for the treatment of resistant TB as Certero, and it was the first novel TB clinical drug in 40 years. It belongs to the diaquinoline class, made up of these two diaryl and quinoline building blocks. And it has a novel mechanism of action in which it selectively inhibits the adenosine triphosphate, or ATP synthase of MTB. It has shown excellent activity against both drug-sensitive and drug-resistant TB strains. However, toxicity has been observed due to its long terminal half-life and home liability that leads to cardiac toxicity. FDA has also issued bedoxolone with a black box warning, which is associated with a serious adverse reaction or death in relation to its usage. Because of the significant side effects, second generation analogs of bedoxolone have been developed. In this work, which was a collaborative project between the TB Alliance and the Auckland Cancer Society Research Centre, over 900 analogs of these second generation analogs were initially prepared. And of these, the top compounds were TVAJ587 and TVAJ876. In animal models, TVAJ876 showed a higher efficacy against MTB in comparison to both bedoxolone and TVAJ587. As of 2020, both the compounds have entered phase 1 human clinical trials. And as of October 2023, TVAJ876 has also entered phase 2 human clinical trials. So there are several issues associated with TB drugs and drug delivery. So the TB granulomas have abnormal vasculature, which compromises the effective delivery of oxygen and anti-TB drugs. And there is evidence that suggests that bedoxolone may not penetrate the necrotic regions of the TB granuloma, instead accumulating in the cellular lesions rather than the hypoxic regions. A potential solution to this is to use hypoxia-activated prodrugs, or HAPs, to selectively deliver the drugs to these hypoxic regions. Prodrugs are generally masked or inert forms of active drugs that are activated in the body, and they're used in cancer therapy cancer drug discovery to increase selectivity and reduce toxicity. It was published very recently that bedoxolone and TVAJ587 were both reported to inhibit human ATP synthase, and the usage of these prodrugs may reduce the amount of compounds free to bind to human ATP synthase. TB granuloma lesions evolve very similar to cancer lesions over time, and under hypoxia, MTB has reduced susceptibility to standard anti-TB drugs, but increased susceptibility to nitronazole drugs. An example of a nitronazole drug is metronidazole, which has been shown to reduce MTB in anaerobic conditions. Pimonidazole and mesonidazole are imaging agents that are used for tumor hypoxia, and their hypoxic selectivity and effective tumor penetration abilities provide rationale for usage of similar compounds in the hypoxia-activated prodrugs. So the overall aim of my project was to optimize these diacronolines for treatment of latent or non-latent, non-replicating TB through application of a prodrug approach to target the hypoxic environment present in the TB granulomas. In this work, I aim to synthesize and characterize novel hypoxia-activated nitronazole prodrugs of bedoxolone, TVAJ587, and TVAJ876, and test them for anti-TB activity. For this work, new nitronazole triggers were also prepared with an aim to influence the prodrug fragmentation rate and hypoxia selectivity. So here is a representation of a proposed mechanism of action of these compounds. So we have an inactive prodrug in normoxic conditions, which undergo bioreduction by endogenous nitron reductases present in these hypoxic environments. In the granuloma, in the hypoxic granuloma, the prodrug will hopefully undergo fragmentation of and release the active drug, as well as the inactive metabolite of the nitronazole trigger. So in terms of the chemistry towards these prodrugs, initially the 3 diacronolines were synthesized according to published procedures. Additionally, 6 nitramidazole triggers were also synthesized, as shown here. By varying the substituents at the N2 position, the reduction potential of the nitronazole can be modified, and this will affect the overall fragmentation rate of the prodrugs. These 2 components were then combined using optimized synthetic groups to form the actual hypoxia-activated prodrug. The compounds were sent for biological testing at the University of Illinois, Chicago, by Dr. Scott Franzblau and his team to undergo the MARBA and LAURA testing, which are standard TB testing assays. Microplate Alamar Blue assay, or the MARBA assay, is used to determine inhibition of actively growing MTB under aerobic conditions, while the low oxygen recovery assay, or the LAURA assay, is used to test compound activity against non-replicating MTB under hypoxic conditions. The compounds were also sent for Bero screening, which screens the compounds for cytotoxicity in mammalian cells. So here is one series of initial biological results we have received for one of the diacronolines, with these triggers. The Vera assay for these compounds showed a similar cytotoxicity in comparison to the diquinoline parent compound. The MAVA and the LoRa results, for the MAVA results, it shows a 3 to 5 times reduction in activity in comparison to the parent compound, while the LoRa assay showed 2 to 3 times reduction in activity in comparison to the parent compound. So these results imply that the activation of the hypoxia activated prodrugs occur in the MAVA assay, as the compounds appear to not be reduced in Limoxia. Under hypoxia, we expect reduction of the nitronazole trigger to occur and activate the prodrugs, and these results, hence this may be happening as we see similar LoRa assay results to the parent drugs. So this suggests a hypoxia dependency of the prodrugs. So this table shows the 2 first-line drugs currently used to treat TB, rifampicin and isoniazid, and the MIC90 results. So both the MAVA and LoRa results for the prodrugs appear to be quite comparable to these 2 compounds, which show some efficacy towards the NTB. In conclusion, 36 prodrugs of Bedoxolin, TBHA587 and TBHA876 are synthesized, and these were sent for biological testing. So these initial biological results demonstrate these prodrugs show some selectivity towards NTB under hypoxic conditions, and this highlights evidence of selective hypoxia-mediated release. Sorry, just one more slide. Future work towards my project, fermentation studies of prodrugs by post-radiolysis with Professor Bob Anderson at the University of Auckland is planned, as well as further biological testing of these compounds in our intramacrophage mouse infection models. And these results will then be used to build and study SARs of the prodrugs, and the development of a prodrug approach for treatment of TB may lead to reduction in treatment time and increased specificity of some existing anti-TB compounds. And I'll just leave this slide up, but I would like to express my thanks to my supervisors and all the chemists at the Auckland Cancer Society Research Centre, as well as all the technicians and our collaborators for the biological testing. Yeah, just open taking questions now.
Great. Thank you for the wonderful talk. If you have any question, please type in the Q&A or chat box. I haven't seen any question yet, so I might start one. So I might miss that, but maybe you have mentioned. I'm just wondering about the mechanism of action for this beta-quinoline.
Yeah.
Yeah. So like, how does it kill bacteria?
Sorry, I can't quite hear you.
Yeah, sorry. So I was just asking the mechanism of action for the beta-quinoline. Maybe you have mentioned, but I missed that.
Oh, yes. So bedoxolin and all the diquinolines, they inhibit the adenosine triphosphate of the ATP synthase of NTB. So, yeah.
Yeah, great. Thank you. Then here is a question from Sam. So how does BDQ compare to other drugs usually used for TB with respect to available and marketability? Availability and marketability.
So bedoxolin is currently available as an add-on agent. So it's always used in combined therapy. So it is not a standalone compound, and with availability. So I'm just a little bit unsure what that, do you mean like how available it is to patients, or?
Yes, it's probably, maybe she was referring to like how competitive or how other drugs compared to other drugs, whether it's relatively available or it has advantages for marketing, I guess.
No, it is currently available to very widely available for treatment. And yeah, so I just didn't quite understand the question.
Yeah, it's okay. Yeah. I think in the interest of time, we might leave there. Thank you, Christine, for the wonderful talk.
Thank you.
Yeah. And if you could share the screen, that'd be great. Okay, great. And our next speaker is Alice Terrio. So Alice is going to talk about mechanism-based modeling predicts antibiotic effect on Pseudomonas aeruginosa, where PK and PD indices cannot. If Alice could share the screen, that would be great.
Fantastic. Can you see that?
Yes, I can see that. Thank you. All yours.
Awesome. Thank you. Thank you so much for the introduction. There we go. So the strain of bacteria that I was working with or the pathogen I was working with was Pseudomonas aeruginosa, which we know causes a range of different infections, including septicemia, pneumonia, and UTIs. It's classed as a priority pathogen according to the World Health Organization, and in 2019 it was associated with over 300,000 deaths. Increasingly, that we're seeing that it's multi-drug resistant, and this is a really big concern, and we need to improve the way that we're treating these infections to try and minimize this and increase successful treatments. One of the reasons that it's so commonly multi-drug resistant is it has a range of different resistance mechanisms that it can use to overcome antibiotic treatment. One such resistance mechanism is the loss of these OPR-D1 entry porins. So when there's less of these porins, less of the antibiotic can enter the cell where it can have an effect. Another mechanism is increased efflux pumps. So when the antibiotic does get into the cell, it's then removed via these efflux pumps. And the third method that I was looking at is increased beta-lactamases, which are an enzyme that can break down beta-lactam antibiotics. And these are three resistance mechanisms that are commonly seen in treatment-resistant Pseudomonas aeruginosa. So the research that I was doing was actually working with 7 bacterial strains. The first one was PA01, a wild-type reference strain. All of the other strains that I worked with from there were isogenic. So they all had specific mutations, but other than those mutations, they were the same as the wild type. So I had strains with a single mutation, one of each of what I just spoke about, and then strains that had 2 of each of those resistance mutations. So they had double mutations. Now, to quickly talk about traditional PK/PD indices, these are indices that are used to link the unbound concentration of antibiotic over time and the MIC to bacterial response. And this has been a traditional approach that's been used for a while. However, we know that they're not accurately predicting bacterial responses, in particular that they can't account for combination therapies, which we're using more commonly. I was working with 2 antibiotics. So I was working with ciprofloxacin, a fluoroquinolone, and we know that it's impacted by increased efflux pumps of the 3 resistance mechanisms that I mentioned. And it follows the PK/PD index of the AUC, the area under the curve, divided by the MIC, or the minimum inhibitory concentration. The second antibiotic I was working with was meropenem, which is a beta-lactam, and we know that it's impacted by all 3 of the resistance mechanisms that I've mentioned. And its PK/PD indice is the percentage of time over MIC. So the first thing that I had to determine for each of these strains was their minimum inhibitory concentrations. And as you can see here for ciprofloxacin, most of them are susceptible. So I've color-coded this as susceptible in green, orange is intermediate, and red is resistant. So we had 1 for ciprofloxacin that was intermediate. For meropenem, there was a bit more of a mix. I then treated them in monotherapy with ciprofloxacin or meropenem. And so what these graphs are showing here is the concentration of bacteria over time. Each of the lines is one of the bacterial strains. And what I really want to emphasize here is that in the top row, I have ciprofloxacin, in the second row, I have meropenem, and these results are standard by the AUC over MIC or the multiple of MIC. And if these PK/PD indices were working really well, we would expect that when we treat all of our strains with the same AUC over MIC of ciprofloxacin, we'd get the same results. But what we can see here is that we got quite different results. So if the traditional PK/PD indices are failing to predict the bacterial response, what can? And this is where I set to work developing a mechanism-based mathematical model to try and describe the results we were seeing. And so I ended up with this model, which if I break down, we have our life cycle growth model of the bacteria, where they're replicating. I then have meropenem. And so what we show here is meropenem being dosed into the broth and then making its way to the active site. However, it's being influenced by the 3 resistance mechanisms that I mentioned. So we know that when there's our loss of entry porins, there's less making it to the active site. And with increased beta-lactamases or increased efflux pumps, there's less meropenem at the active site. Similarly, for ciprofloxacin, it's dosed into the broth, but the presence of efflux pumps removes the amount of meropenem at the active site. There's also a synergy function between the concentration of ciprofloxacin and the effect of meropenem. And then the concentrations of ciprofloxacin and meropenem at their respective active sites cause direct killing for the bacteria. Now, I also was running this with 3 subpopulations. So this model represents a double susceptible subpopulation, so it's susceptible to both meropenem and ciprofloxacin. There's also a subpopulation that was intermediate to meropenem and resistant to ciprofloxacin, and vice versa. And what I'm showing you here is the same graphs as before, except for this time the lines aren't just connecting the data points, the lines actually represent the modeled fits. And there's no inter-individual variability here, so they're describing the whole populations that we see. We can also have a look at some of the combination therapies, and there's a lot going on here, but what I would like to highlight quickly, actually no, I'm going to zoom in, because altogether I modeled about 292 replicates, including biological replicates, with the 7 strains and a lot of different concentrations. So I will zoom in onto just a few of our combination therapies for one of the strains. And what you can see here is our monotherapies, which are shown in the blue, the orange, and the green, saw initial killing, but then there was regrowth to the level of the control, which is in black. Whereas the combination therapies, which are in that navy, the purple, and the pink, saw killing and no regrowth. So combinations were successful. And the model was also able to describe that, which was really exciting. So to conclude, the PK/PD indices alone didn't predict the meropenem or ciprofloxacin exposure required to suppress bacterial regrowth over 72 hours. And mechanism-based mathematical modelling, accounting for the different resistance mechanisms, could predict the bacterial response and account for combination therapies. Next steps from this study include dynamic studies to see how these go with changing concentrations and that continual flow of media and antibiotic, and also genomic sequencing to see if there are changes to these strains over time. With that, I'd just like to thank my supervisor, Connie, and my lab group. Thank you.
Thank you. That's a very good talk and also on time very well. Thank you very much. Do the audience have any questions to start? Oh yes. So there are 2. I think I'll probably start with Glenn and then Christine. Also, is it pause? Yep. So I might start one question then. So in terms of a big picture though, like is it possible to include host factors in there? Because I think in the broad in the future, you probably want to apply the system that could be used in clinically. Is it possible to incorporate host factor?
Yeah, so in terms of applying it clinically and then including host factors, it's sort of a 2-pronged approach. For applying it clinically, ideally what you'd be able to do is take a sample from the patient, sequence the genome of that bacteria, and then use that information to help inform the best dosing option. It can be taken a step further to include patient factors, And that I know there's work at the moment that's being done, particularly in the AI space, to start to work out which patient factors are most critical in determining bacterial outcome and the necessary treatments. So there is work that's being done in that space. Yeah.
Yeah, awesome. Good to know that. And here is another question from Sahem. So Sahem is asking, what is the basis for the PK/PD indices for predicting bacterial resistance, and why is it not as accurate?
They haven't been used to predict bacterial resistance. They were used to guide dosing, and so to guide dosing targets, and to sort of say if we can hit this target, we should see successful killing. They were established based on running experiments in mice models and mapping out which of the 3 indices had the best correlation for a certain antibiotic. Why it's not as accurate as we hoped it would be, there's a few things that are at play there. One is that we know that our MIC, so all of the 3 PK/PD indices are linked to the MIC, and our MIC is a singular time point check. So we look at our MIC generally at 24 hours, and that's not really going to show you the full time course. You know, the graphs that I was showing were over 72 hours, and some of them you would look at 24 hours and say, yeah, there's killing happening there, but by the end there's regrowth. So they're not really showing the full picture in that regard. And as I mentioned as well, they also can't accommodate for combination therapies.
Yeah, it's a good point. Yes, I might follow on another question. Like, is there any other antibiotics that you're trying to model or like interested in the future?
Yeah, absolutely. And a few of my colleagues are presenting also in this session, and they'll talk about different antibiotics. So I won't steal what they're presenting, but
Yeah.
There is, and we have the ability to model all different sorts of antibiotics. Yeah.
Yeah. Oh yeah. Oh, there's another question come in. Yeah. Oh, 2 actually. The first one is about Pseudomonas use an efflux pumps to avoid the effects of antibiotics on it. To using targeting efflux pump system is a good strategy to tackle these passages. It's from AVA.
I can't say that I'm an expert in this area to give you a good answer on this question. I mean, it's something that could be considered, and if we can, you know, stop these efflux pumps, then perhaps, yeah, we'll stop the efflux of removal of the antibiotic, which might work. I'm not sure if there's work that's been done in that space. I don't think at the moment there is any antibiotics or any things that are being paired with antibiotics to prevent that, though.
Yeah. All right. And our next question is from To. So is there any reason why you choose Cipro and meropenem for the modeling?
Yeah, so ciprofloxacin and meropenem are both used to treat Pseudomonas aeruginosa. We could see that they're monotherapies, so when they're given by themselves, they're not always effective at killing the bacteria and preventing regrowth. So to look at that combination was interesting and see that it did have successful treatment.
Yeah, right. Yeah. All right. Our last question from Anton. So Anton was interested in we have recently started a beta-lactam therapeutic drug monitoring service at the Alfred. Can we apply MBM to determine optimize the dosing changes rather than simple single point analysis?
I think not yet. Hopefully soon, but realistically not this year. You can't. Yeah.
Yeah. Okay. Yeah, we might leave there. Thank you very much, Alice,
Awesome.
for
Thank
wonderful
you.
talk and the questions answering. Yeah. Our next speaker is Dominika Fu, if hopefully I pronounce your name right. So Dominika is going to present the effects of different resistance mechanisms on Pseudomonas aeruginosa response to meropenem. Yeah, all yours.
Thank you so much for the introduction.
Oh, I think
sorry.
The slides might change. Yeah.
Can you see that now?
It's not in full screen mode. Yes, yes, good.
Can you see it now?
Yes.
Oh, good. And you can hear me?
Yes, I can hear you well. Yes.
Oh, good. Okay, thank you so much for the introduction. So I'll jump straight into it. Okay. So the bacteria that I was working with was Pseudomonas aeruginosa, and this bacteria was associated with more than 45,000 deaths in 2021 alone. One of the drugs that's been used to treat Pseudomonas aeruginosa infections is meropenem. And in order for meropenem to work, it has to get into the bacterial cell wall, into the periplasmic space to exert its mechanism of action and essentially kill our bugs. However, bugs are very clever, and as Alice also mentioned, they can develop resistance mechanisms that greatly reduce effectiveness of our antibiotics, such as meropenem. And those resistance mechanisms can develop even during treatment. And as my colleague already mentioned, bugs such as Pseudomonas aeruginosa can downregulate production of porin channels, which means that meropenem can get into the periplasmic space and work. It can also produce enzymes which break down meropenem, rendering them not working. Or they can upregulate production of efflux pumps that actively pump meropenem out of the periplasmic space, which means that meropenem can't work anymore. And traditionally, we've been using PKPD indices to inform human dosing regimens in the clinic. And for meropenem, the most important one that's used in a clinical practice is the time that the end-bound antibiotic concentration exceeds the minimum inhibitory concentration, MIC, of the infecting pathogen. However, this traditional approach has limitations. For example, there's only one assessment time point, usually at 24 hours, which means that we do not really capture the full-time course of antibiotic pharmacokinetics as well as bacterial response. And we cannot really account for emergence of resistance. So what else can we do to maximize bacterial killing and minimize the emergence of resistance? Well, this is where our project comes into play. We firstly aim to characterize the effects of the various different baseline resistance mechanisms present in Pseudomonas aeruginosa on bacterial killing and any resistance emergence. We wanted to evaluate whether the traditional approach could predict these effects, and we wanted to develop a mechanism-based mathematical model that could describe bacterial outcomes. A novel model that includes key bacterial characteristics that would inform drug effect parameters. And a model that would be able to characterize full-time courses of antibiotic concentrations as well as the total and bacterial resistant bacterial counts. So in order to do that, we conducted hollow fiber infection model experiments using 5 different clinically relevant meropenem dosing regimens and of course controls. We had 3 different continuous infusion meropenem dosing regimens that we've tested, and also 2 different intermittent dosing regimens of meropenem. And just briefly, in a simplified way, hollow fiber infection model experiments are in vitro tests. This is like a system of pumps and tubing and hollow fiber cartridge with microfibers where bacteria actually sits. And in this experiment, we're able to simulate many different antibiotic dosing regimens, various pharmacokinetic drug exposure profiles that we see in humans in real life, and we're able to test various antibiotics and see if they would work or not. So in our experiments that ran for 10 days, we tested 7 different isogenic strains of Pseudomonas aeruginosa. They were all constructed from the PA01 reference strain. And the only difference between them were the resistance mechanisms introduced, which allowed us to directly compare between them. And we had 3 different single knockout strains, which means that that into their genome we introduced one of the resistance mechanisms. And we also had 3 different double knockout strains that had a combination of the 2 resistance mechanisms into the genome, which allowed us to study the complex interplay as well between the different resistance mechanisms. And the model that we developed roughly consists of 3 different parts. First one describing bacterial dynamics by life cycle growth. Another one that describes antimicrobial effect by meropenem, by direct killing effect. And this is where we included and incorporated the effects of these different resistance mechanisms present by themselves, but also in combination. And our holofiber infection model studies, they simulated pharmacokinetic exposure profiles for meropenem in critically ill patients with normal renal function. So the model that we developed was able to characterize all viable counts on drug-free, as well as meropenem-containing plates, agar plates, which represents the resistant bacterial subpopulations across all 7 strains simultaneously, without estimating any strain-specific drug effect parameters. And in our model, for the concentration of meropenem at the site of action, so in the periplasmic space, was dependent on the presence of these different resistance mechanisms in our strains. And this is just a schematic of the model that we've developed. The top part represents the antimicrobial effect by meropenem, and the bottom part describing the bacterial replication and growth. And this is a snapshot of all of the results that we've gathered. Today, I wanted to talk about 3 different strains and 2 different meropenem dosing regimens. So on these graphs, you can see 3 different strains and how bacterial concentration on the y-axis changes with time on the x-axis during our experiment. The points or the symbols are our data points from the experiments, but the lines here are our population model fits without any inter-individual variability between them. So was the traditional approach able to accurately predict bacterial response to treatment? Well, not really. For example, regimens achieving 98% time of one-time MIC for one of our strains suppressed regrowth and resistance of that strain, while even much higher target of 100% time above 5 times MIC failed to do so against 2 other strains, despite these strains having the same MIC. Because in theory, if the traditional approach could predict bacterial outcomes, all of the strains with the same MIC would respond to the similar treatment in a similar way. But that's not what actually happened in real life. However, our model, the one that we developed, very well characterized bacterial response for the total bacterial populations, as you can see here, represented by the model fits, but also resistant bacteria, as you can see here on the meropenem-containing plates, which means that the model was also able to characterize resistance, emergence of resistance. So to conclude, what we saw was that the pharmacodynamic response was much more complex than it could have been predicted by the traditional approach itself. Even for strains with the same MIC, based on resistance mechanisms affected bacterial outcomes. And models such as mine or Alice's provides better understanding of the complicated relationship between antibiotics, bacterial characteristics, and resistance emergence during treatment. Models such as these are a first step towards personalized medicine, not only personalized towards the patients that's infected by the pathogen, but also taking into account the certain characteristics of pathogens as well. Thank you.
All right, thank you. In the interest of time, I might have just a quick question. Is there any question that you want to ask? Please put it in the Q&A or chat. All right. So if not, I'll just ask a quick question. I'm just wondering, like, in terms of resistance mechanisms, do you have to know the resistance mechanism every time when you do the modeling?
Yeah. So our model, for models such as this, when we looked at the resistance mechanisms, we do have to know what resistance mechanisms bugs have. But another thing we can do is that at the end of our experiments, we can do genomic analysis to see if any new resistance mechanisms popped up. And our model can be updated to account for also changes in bacterial genome or resistance mechanisms expression during treatment.
Okay. Yeah, thank you very much. We might move on. So our next speaker is Jamia Hemphill, and Jamia is going to present investigating antimicrobial activity in the human gastrointestinal microbiome. If you could share your screen, that would be great.
Yep, sorry, bear with me a second.
Yeah.
All right, that looking
Yeah, I can see you. Yes, all good. Yeah, I can hear you well.
Awesome. Thank you for the opportunity to present today on my project looking at antimicrobial activity in the gut microbiome. So most bacteria reside within communities of many different bacterial species and strains where competition is really high. And so some bacteria, in order to gain a competitive advantage, produce antimicrobial molecules that can target their bacterial neighbors. So as Mark just told us before, a lot of our current clinical antibiotics are derived from soil bacteria from research done in the 50s. And we've been now looking at different environments as well. So communities in marine environments, Antarctica, and more recently, the human microbiome in order to discover novel antimicrobials. And so we are particularly interested in looking at the gut microbiome as the communities within there are very dense and diverse and competition is really, really high. And so they're going to need to have some kind of advantage, which could be antimicrobial production. Additionally, they are naturally in competition with human pathogens. So these may be opportunistic pathogens that reside within a healthy gut normally, such as C. difficile, or foodborne pathogens that pass transiently through, such as Salmonella and E. coli. And so our gut microbiota may have specific antimicrobials that can target these pathogens. So far, there's been a lot of metagenomic data that has identified novel biosynthetic gene clusters that could encode antimicrobials. However, this data is limiting as it's only able to identify genes that are similar to known antimicrobials. And even then, even if there is genomic similarity, it does not necessarily guarantee that there's going to be functional similarity. And so hence why it's really important to be able to culture these bacteria and perform these phenotypic assays to confirm this antimicrobial activity and then perform further assays to understand their function within the gut. And so not only am I interested in the production of antimicrobials in the gut microbiome, but I also want to investigate the role that they play within the microbiota community. So if we introduce a new therapeutic, such as a new antibiotic or a live biotherapeutic, what's it going to do to these communities? Is it going to wipe them out, or do they have their own immunity from naturally living alongside these strains in the gut normally? And then additionally, how does the host get involved there as well? Is it going to be toxic to the host? Are they going to release their own things that may impact these communities in a different manner as well in response to these antimicrobials? For today, I'm just focusing on those first two, so the antimicrobials themselves and their role in the community. So my first aim is to phenotypically and genomically screen bacterial members of the gut microbiome for antimicrobial activity, and then to characterize the functional role of these antimicrobials within our commensal communities in the gut. So for the first aim, I conducted a high throughput agar overlay assay screen. And so I screened 284 commensal gut isolates by culturing them on agar plates and then overlaying that growth with agar that had been inoculated with one of my multi-drug resistant gastrointestinal pathogen strains. So we have C. difficile, E. faecium, E. coli, and Klebsiella. And then you can see in this example here, observing those for signs of inhibition that we can see in this clear zone around that commensal isolate, indicating that it's inhibiting this pathogen that is growing over the top. And so this phylogenetic tree here shows the results from that screen. So the commensals are represented in the tree, the branches coloured by their phyla. So we've got the firmicutes in blue, Bacteroidetes in green, Actinobacteria in yellow, and Proteobacteria in red, with a few other phyla in there. And then the bar graph around the outside indicates which of these pathogen strains each of those isolates exhibited inhibition from. And so 23% of the commensal isolates exhibited inhibition of at least one pathogen strain. But as you can see here, many of them were actually showing inhibition of more than one strain. So the next step was to select 19 of these inhibitory candidates for further investigation. And so they're just represented here with their inhibitory phenotype in the bar graph. And so then I screened the genomes of these 19 candidates for any genes with a high similarity to known antimicrobial genes. And interestingly, only 3 of these 19 candidates returned a match for an antimicrobial gene that's already been characterized. And so the disparity between this inhibitory phenotype and the antimicrobial genotype that we're seeing really highlights the importance that we of being able to perform these phenotypic assays to investigate antimicrobial activity. And so next, in order to establish a model to investigate the role of these antimicrobials within our gut commensal communities, I selected one of these identified genes, so enterolysine A, produced by this commensal Enterococcus species. So enterolysine A is an antimicrobial protein with bactericidal against gram-positive bacteria through the action of degrading the cell wall. And it's been previously shown to inhibit Enterococcus species, Pediococcus species, Lactococcus, and Lactobacillus as well. And so, I cloned enterolysine A into an E. coli expression model and then tested that expression model through this co-culture system. So I'll quickly run through it with you. So here we have what we call a duet that has 2 wells, and they're separated by a semi-permeable membrane. So in one well, we inoculate the transformant E. coli that's producing enterolysine A, and in the other well, we inoculate the pathogenic Enterococcus faecium. The bacterial cells cannot pass through this membrane, but any metabolites, proteins, or peptides that they are producing can pass through. And so this would include enterolysine A. And then the growth of the bacteria is measured through taking optical density readings. And so here I'm showing the growth of those pathogens. So in black is when it was opposite from the E. coli with the empty vector, so no enterolysine A, and in pink is when enterolysine A is being produced. And as we can see, usually at around the 36-hour mark, enterolysine A starts to inhibit this Enterococcus pathogen, showing that my expression model was successful, and enterolysine A does still have this antimicrobial activity that we saw in the original commensal isolate. So in summary, for enterolysine A, I was able to phenotypically identify commensal gut isolates with antimicrobial activity, also genomically identified isolates with potential antimicrobials, cloned enterolysine A into E. coli, and showed that it was able to inhibit pathogen growth. So the next step, I wanted to look at what role enterolysine A plays within commensal communities. And so in order to do this, I set up this assay where we've got co-culture in broth of the E. coli transformant expressed in enterolysine A with a commensal community, and then comparing that to the E. coli empty vector. So here is the community that I've constructed, and it's designed to be a smaller mock version of what we would see in a healthy microbiome. So the majority are Bacteroidetes and Firmicute species with an Actinobacteria, and then the E. coli making up that Proteobacteria. And I've just highlighted the gram stain of each of these isolates. As I mentioned previously, enterolysine A affects gram-positive bacteria, and so that's where I'm expecting to see most of the change here is in these gram-positive isolates. So samples are taken at each of these time points over 72 hours, and at each time point we're looking at total cell count changes through optical density readings and CFU counts, then looking at species-specific changes through qPCR and metagenomics, and then any functional changes within the community through transcriptomics as well. So here is a heat map just showing the species-specific response to enterocolisin A in the community. Now, just bear in mind, this is only N of 1, so I'm not going to make any conclusive statements here. But what we're seeing here is the red indicates that the proportion that that specific species made up in the community was reduced when enterocolisin A was present, and blue indicates that it increased and made up a larger proportion in the community when enterocolisin A was present. So already we're seeing that different species are having different responses at different time points as well over the 3-day experiment. So this Enterocloster species here we can see in the first 24 hours it seems to be reduced by enterocolisin A, however it then recovers in the last 48 hours. And then Lactococcus almost has an opposite effect where it thrives in the first 24 hours but then is reduced within the community. So in summary, I have so far demonstrated that enterocolisin A can inhibit pathogens and also has a varied impact on different species within a commensal community. So it does have the ability to change our commensal communities as well. So to give this a broader context, this community model will enable us to investigate how antimicrobials facilitate interactions within the commensal communities. And then we can gain an understanding of how the microbiome may respond to novel therapeutics that are going to try and prevent antimicrobial resistance. I would just like to acknowledge my lab for all their support, and as well as the other people that have provided plasmids and pathogen strains as well. Thank you so much for listening.
Thank you, Jamia, for a wonderful talk. I can see there is a question already. So this is from To. Is the enterocolisin A gene only possessed by E. coli, or can this gene be transferred to other organisms?
So it's only naturally found in Enterococcus species, specifically Enterococcus faecalis. I haven't looked at if it can be transferred to other species within the gut microbiome, but I think that it is a specific Enterococcus gene.
Yeah, thank you. And the next question is from Bihana. Sorry if I pronounced your name wrong. Is the media rich, maybe not triggering the expression of the antimicrobial peptides so much?
Yeah, so we use a really complex media called YCFA that we've designed specifically to grow the quite fussy gut microbes. So it is actually very rich, and I think it is triggering the expression of antimicrobial peptides. I think the reason that we're seeing like there wasn't a massive reduction in the growth of the pathogen is because the E. coli expression model isn't as efficient at producing the enterocolisin A, and so I'm actually also working on creating a knockout mutant because I believe that the wild type Enterococcus will have a greater effect within this community, and it'll be interesting to compare the two as well.
Thank you. And then the next question is from Abia. So Abia is asking, thank you for your interesting talk. Given that the gut microbiota carry AMR genes, the gut resistome, could they serve as a source of resistance for opportunistic pathogens like C. difficile?
Yeah, that's not really what I focus on, but there's definitely been a lot of research into the transfer of AMR genes from the gut microbiota to pathogens, and I do believe it does occur, unfortunately.
Yeah. And then there's a question from Sabrina. Great talk, Jamie. Is there a reason known that different gene species respond to and enterocolisin A differently?
That's a really good question. I think so. Interleucin A is a bacteriocin, and so far, what we know about bacteriocins is they usually target species that are quite closely related to the producing species. So it could be, the specific mechanism in which it targets it and also is able to enter the cell is reliant on how closely related and similar it is to that producing species. Again, there's that difference between the gram-positive and the gram-negatives. But there's not a lot of research into this specific enteroleucin, so I'm not super sure.
Yeah. Okay. So then the last question from Sam. So great talk. With respect to the changes in bacterial communities due to enteroleucin A, do these changes reflect to any potential leading issues or betterment of the host? In other words, are the species increasing beneficial or harmful?
Fantastic question. Again, that's the whole idea of my PhD and what I'm really interested in looking at. Because if we're seeing that it's changing these communities, it could also be used in a different way. There's a lot of research at the moment looking at modifying the microbiome for not just antimicrobial resistance, but for specific diseases. So if we see it's targeting a species that has been known to have associations with cancer, we could use this to alter that. Additionally, if we discover that it's really harmful to what are generally considered as the good bacteria, you probably don't want to use it as a therapeutic, and you'd probably want to pursue a different antimicrobial.
Yeah. Thank you very much, Jamie. We're looking forward to your PhD work in the future. Yeah, I'll leave you there.
Thank you so much.
Yeah. And I'm going to hand over this session to my co-chair, Glenn.
Thanks, Jihan. The talks have been amazing so far. Our next speaker is Siobhan Breen, and she'll be talking on Tazobactam plus meropenem against ST235 clone Pseudomonas aeruginosa isolates in hollow fiber infection models. Take it away, Siobhan. Thank you.
Great. Thank you so much for that introduction. Can you all hear me okay? Yes, okay, great. So I'll just jump right into it today. So Pseudomonas aeruginosa is a pathogen classified by the World Health Organization as high priority pathogen, and this is due to its extensive range of antibiotic resistant mechanisms, which you have heard about today from my colleagues. Antibiotic resistance can be present in a bacterial population, but as a subpopulation, and this is due to random mutations that just occur. Administration of an antibiotic may not kill our total population and not kill this resistant subpopulation, and therefore this resistant subpopulation can amplify and soon become the total population for future treatments, which means this antibiotic would not be effective. We then thus need an urgent change in the way that we dose our antibiotics so that we stop having these suboptimal treatment options and things happening, causing a greater clinical problem. So Pseudomonas aeruginosa is also a huge problem because it has different clones, and clones are just bacterial strains which are grouped together by their common genomic properties. Pseudomonas aeruginosa has many, but 3 of them have been classified as high risk, and they are here ST111, ST235, and ST175. And the issue with ST235 clone, which is the one I focused on today with my experiments I'll be showing you, is that the strains are really well known for being extensively drug resistant and hypovirulent, as well as being a main cause of endemics worldwide, especially in hospitals and for hospitalized patients, which again shows us that this is really a huge clinical problem that we need to address. So my experiments aim to look at 2 very commonly utilized antibiotics, ceftolozane tazobactam and meropenem, against ST235 strains which were collected from patients in hospital. We wanted to first evaluate the time course of bacterial killing and regrowth in an in vitro dynamic system. And then we wanted to investigate this resistome of the genomic of the bacteria and look at the genomic changes that may have occurred in these bacterial strains over treatment. And lastly, we wanted to develop a novel mathematical model to describe this data. So as you've already seen today, this is what a hollow fiber infection model looks like. I will go through it quite quickly, but this is what we use to mimic a human infection with our bacteria. So we have our inflow media, which pumps new media into the central reservoir, where we then mix it with our antibiotic before it then goes up into our cartridge, which is holding our bacteria. And this cartridge has pores, which lets our fresh media in and drug in, but our old media and drug out to return to the central reservoir before being removed from the system. And in this experiment, we used the approved dosing regimens of our antibiotics, and the doses we used were the achieved antibiotic concentrations seen in population pharmacokinetic models. In this experiment, we had continuous infusion meropenem and intermittent ceftolozane tazobactam. And you can see that we were able to achieve our targeted profiles for both of our antibiotics. This graph here is showing us our ceftolozane concentration for the experiments over time, where we can see the targeted profiles in the black lines here, with our symbols indicating the ceftolozane concentrations from the experiments. And here my red dotted line is what I modeled the fits to be. So we see this all fits quite well. For my genomic sampling, we wanted to determine any changes that occurred to our bacteria through time. And so we took samples before and after treatment of our antibiotics. And when we took the samples, we then broke them down so that we could have just the DNA, and then we sequenced them to really look at the sequence and be able to ask ourselves, why did the bacteria regrow here? And lastly, the last thing that I did was create a mathematical model which comprised of 3 different sections: our bacterial dynamics, our antimicrobial effect, and of course our pharmacokinetics of our antibiotics. So we had our growth cycle model for describing Pseudomonas aeruginosa growth and replication. Then when I had my meropenem, there was a direct killing effect, whereas ceftolozane was described as inhibition of successful replication. And we only described the differences between the different treatments and the strains with the resistant mechanisms that were present through treatment or before treatment, which is something that I'll go into a bit more depth a bit later. Our model also included the hollow fiber concentrations that we had for our antibiotics. So this is what my data looks like. Here you can see I had 3 different strains, ST23505, 09, and 10. And here I'm showing you our total viable count, so everything that grew on an agar plate. And we have the log CFU per mil, so basically the amount of bacteria over time here. And firstly, at baseline, we can see that all of my strains had an OPRD mutation, and they also had an, the OPRD mutation affects our meropenem influx, and they also all had a mutation in either AMP-R or AMP-D, which increases the production of enzymes which break down both ceftolozane and meropenem. So if I start going through the data, we can see here the control in black, it performs as expected and plateaus. We can then see that the meropenem monotherapy failed to suppress regrowth for all of our strains. And we see here that ceftolozane tazobactam worked as the same as our meropenem when we look at it against ST23505 and 09. However, when we look at it against ST23510, it was successful at inhibiting our regrowth. In all cases, however, we did see that the combination was successful, and it did actually perform synergistically against ST23505 and 9, meaning that it had a greater than 2 log decrease from the initial inoculum and our best performing monotherapy. If we look at ST23510, we can see here that the combination was able to reach the limit of quantification much quicker than the ceftolozane tazobactam monotherapy. And also just to note here, again, we do see that my experimental data is the symbols, but my lines here are the mathematical model fits that I was telling you about. These are my population model fits, meaning that there's no inter-individual variability between the different treatments. So we can also look at our resistant subpopulation by growing bacteria on plates that contain the antibiotic. Here we're looking at my meropenem resistant subpopulation. And what we see here is that our control in the black line again shows us the basal level of resistance. So just the normal amount of resistance in a population. However, when we look at our meropenem monotherapy, we actually see that it grew higher than the control, meaning that resistance was amplified through the treatment with monotherapy meropenem. And actually in our mathematical model and also just through our genomic studies, we saw that there was an FTSI mutation here, which is a mutation at PBP3, which is the main location for binding for meropenem to have its effect. So we see that this mutation occurred, and this is most likely what caused this amplification of resistance. When we look at our combination, you see here that the counts are all lower than that of the control, meaning that it was resistance suppressing. When we look at our ceftolozane tazobactam subpopulations, we see basically the same trends here, where the resistant regrowth of the ceftolozane tazobactam monotherapy was seen to be caused by an AMPC mutation, which again leads to hyperproduction of our beta-lactamases, which affects ceftolozane now. And of course, in ST23510, we don't see this mutation, and we didn't see any resistant regrowth because the monotherapy was effective. So if we look at our genomic data obtained in these experiments, we used these as the only parameters in our mathematical model to differentiate the model fits. And as you saw just before, it described all of our data very well. This is the first time that something like this has been done for isolates from patients. And as I mentioned earlier, we had our OPRD mutation before treatment. So this was in all of our isolates to decrease meropenem entry to the bacteria. We also had that AMP-R or AMP-D mutation at baseline in the model to describe the enzyme production to affect both meropenem and ceftolozane. And then we included the FTSI mutation only to the meropenem resistant subpopulation in my model to describe why this meropenem monotherapy regrew. And then, of course, we had our AMP-C mutation as well, which was applied to ceftolozane resistant subpopulation only. And so again, these genomic differences were the only thing required in my model for it to fit my data. So finally, I'd just like to conclude by explaining that ceftolozane tazobactam monotherapy was effective in one of our 3 strains, whereas my meropenem monotherapy was never effective. But if we look at my combination, it was always effective against these strains, and my novel mathematical model was able to characterize this data utilizing the genomic information. And so this is really a step towards personalized medicines, not only looking at patient characteristics, but also really looking at the affecting pathogen in this case. So lastly, I'd like to acknowledge my team and thank you.
Thanks so much, Siobhan. Are there any questions? Anyone? No? Okay. Well, while people are writing them down, I had a few. I thought the hollow fiber model was really cool. That's a really cool model. I was wondering, in the host, there are a lot of factors that aren't present in vivo. You've got your immune system and host signaling factors and things of that nature. Do you have any idea how you could incorporate some of those factors into your model, and do you think you would get different outcomes?
Yeah, I think a really good question, and something we definitely are trying to include in our model to, help these in vitro studies. As of yet, there's not too much that we have found that is useful to add. The main thing is that, with these in vitro models, we're able to sort of look at the worst case scenario. So if we do have a patient that has very limited or no immune system at all at this point, we can at least see if the antibiotics will be able to sort of get us to a much better level of bacteria in the patient. But yeah, also, like I mentioned with ST235 isolates, they are all considered quite hypervirulent. So actually with a mouse model, testing that was done recently, it showed that the immune system had little to not really any effect on the ability for the patient to get better. So hopefully by sort of having this model, it allows us to sort of still get some insight.
Yeah. Paul has a question. Do you think there is another resistance mechanism for the combination?
Yes, so we did see in one of our strains when we did the genomics, we saw that there was this ability for the bacteria to to delete a huge section of its genome, which included all of its MEX X, Y, and Z, and as well as the GAL-U region. And so this really puts the bacteria in a state of being pretty non-functional and in a very low metabolic state. But it was able to then become resistant to the antibiotics. However, in saying that, when we did see that resistance occurred after 200 hours of treatment, whereas the resistance we saw to the monotherapies occurred within 24 to 72 hours of treatment. So we're at least giving the patient a much larger window to be able to get better in this case.
Cool. And there's just one last question from Huai. She says, or they say, really interesting talk. Have you considered acquired antimicrobial resistance genes when explaining AMR mechanisms in those ST235 isolates?
Yeah, definitely we have looked at them. All the strains I looked at had no acquired resistant genes. They only had like the standard resistant genes that it had. But definitely I've got some colleagues working on some strains that have some acquired beta-lactamases and different resistant mechanisms like that.
Fantastic. All right. I think Siobhan has fielded enough questions, so thanks very much, Siobhan.
Thank
Our
you.
next speaker is Tope Ibsanami, and he'll be talking on the comprehensive resistance factor profiling and data-driven computational drug discovery targeting virulence factor regulators from whole genome sequencing analysis of Pseudomonas aeruginosa. That is quite the title. Please take it away, Tope. You're welcome to share your screen. So I can't hear you, Tope, I don't think. Yeah, I think you might be muted.
Can you hear me now?
Yes, can hear you now.
Oh,
Fantastic.
sorry about that.
That's all right.
Yeah, it's upon us today. And
Oh, and sorry, you've got presenter mode shared, so you want to share the other screen.
okay. Is it better now?
Yeah, that's fantastic.
Oh, sorry about that. Yeah, so antimicrobial resistance causes a lot of money and it's predicted to like kill more than cancer by 2050. Assuming that it's not bad enough, antimicrobial resistance, antibiotics have also been used in healthcare. We also use them in veterinary and livestock and the likes, which what we thought was contained as healthcare problem is now affecting our entire food chain, which makes this a global challenge for us. And one thing we can create an urgent need for development of novel antibiotics treatment. Another way you can fight them is creating awareness and some innovative programs like this. P. aeruginosa is Pseudomonas aeruginosa is one of the major hospital-acquired infection and is a paradigm of antimicrobial resistance due to the multi-drug resistant mechanism. and some of it is this large genomics. One aim of this project is to gain more genetic insight into the pure genocidal sequence and compare the newly isolated sequence to those old ones to see if there's a new development in their genetic makeup or what are the reasons why those new isolates have become more resistant. Another important thing that we want to check is one of the mechanism of resistance of PQNR, pure genocide, which is QS interplay, which is quorum sensing. And the most important receptor here is MVFR, and it can also be called PQSR. MVFR is the multifactor virulence receptor. The reason why this one is important is because it's working downstream of LAS-R and also upstream of RLX-R, which make it very important to drug target. So the concept here is that if you're able to develop a drug or identify a drug that will be able to inhibit this, it will have a great overall effect on the QSR interface. So these are one of the approaches we use on this project. We're able to get our genomic sequence of pure genocidal from NCBR, then we carry out genomics analysis on it. From there, we're able to like identify genomic sequence of PQSR, which is one of our interests on this project. From there, we carry out our AI modeling to predict the 3D structure, and we're able to predict the binding buckets from there. Another aim of this project is one of the most problem in medicinal chemistry is like determine which analog of drug are we going to synthesize, which one will be very effective in the lab. And also, like Professor Mark said earlier, the issue of funding in drug discovery. And a way we can address that is by using bioinformatics approach. So here we did a pharmacophore modeling to identify 8,200 compounds from our database. Then we proceed with molecular docking, molecular dynamic stimulation, and we'll be doing biological testing and lead compound optimization very soon. So from the genomics data analysis, this is a subsystem that we're able to get from there. So a subsystem is a set of proteins that together implement a specific biology process or a structural complex. So from there, we move further to get their specialty gene. This is the number of gene, then the specific source from different database. So here we have VFDB database and a lot of other database. Then we move ahead to like classify some of those resistant genes into different mechanism of action. Then we're able to get the one under efflux pump, under antimicrobial target replacement, and different other resistance mechanisms. Also, to address one of the aim of this project is to see if those newly isolates of pure genocide, if their genetic makeup is a little bit different or there's deviation from those ones sequenced before in public database. So we carry out phylogenetic analysis on the PQSR or MVFR we isolated from our genomics analysis, and we're able to compare with some of the homologous present in PDB database. And you can see for edge, we found out that 69% relationship with 6q74. This 69% might seem like a small variation or deviation from this sequence, but we never can tell. Those small variations can also matter and might be influencing the genetic made up of those resistance. Then further, the other part of this project is molecular docking. So from the 8,200 and 8,025 compound we got from SIN database, we carry out molecular docking on them. We divided into 4 degree groups, group A, group B, group C. So the result is shown here, it's the molecular docking result, and it's measured in kilocal per mole. To interpret it is actually very simple. The concept is that the more negative the result is, the better the binding affinity, and the better the binding affinity, the more effective the drug is. So from this section, the most active one is compound A1, and this is the ID of the compound in case you want to check it on a database. This is the results, scatter plot result of the top 10 compound from the group B. Also the same concept in interpreted in. Same as group C and group D. So after that, we can go on to like check the 2D interaction to see how the drugs bind to that particular receptor and different bond that was formed. We found out that there's a lot of hydrogen bond, and literature review in the past have shown like hydrogen bond are also very effective in and also very important in drug discovery and compound receptor binding. Then also with there's the compound from the top compound B1 from group B. I also find out there's pi stacking bond with tyrosine here, and similar to the one of the most active drugs with this receptor M64, which form a pi stacking interaction with tyrosine 258. Same as this, a lot of hydrogen bond interaction and this. So further, we proceed to molecular dynamic stimulation, because molecular docking is not enough for us to determine whether a drug is active or not. There's a bunch of other tests to do, like Admens profiling, binding affinity tests. So one of the tests we did is molecular dynamic stimulation, which involves stimulating the molecule of an atoms over time and give a molecular dynamics relationship of it. So we go on to plot the RMSD plot, which measures the average deviation. This measures the average deviation of a particular protein ligand complex. The interpretation is that it shows how stable a protein ligand is. So here it's the equilibration state, and here it's during the stimulation. And to interpret this, we actually check whether it's over any value that is below 25 angstrom or 2.5 nanometer. It's good according to like previous literature. And we also check if there's a high or fluctuation, or whether there's a great peak in it. So those are the two things we check for: how high the value of our RMSD is and the number of fluctuation we're able to get from there. So we also go ahead to plot the radius of gyration, which also similar interpretation. Conclusion. This study showcases how advances in computational power artificial intelligence strategy can be incorporated in drug discovery, contributing to the development of target and selective new therapeutics, while also lowering the expenses of drug testing and drug discovery. Acknowledgement. Thank you.
Cool. Thanks, Tope. That was a great talk. We have already a question from Tor. Tor says, great talk, Tope. Given that the bacterial regulators can be in a complex interaction with others, e.g. two-component systems, how do you take that into account into your model, or do you take it into account?
Oh, can you repeat the question again, please?
Yes. Sorry. Given that the bacterial regulators can be in a complex interaction with others, e.g. two-component systems, do you take it into account into your model?
Oh, yes, we also consider that, like in one of my presentations, I mentioned, especially for this, I mentioned that the MVFR, which is also PQSR, have an interplay in other receptor, which is also make it a important target in the complex of the QSR. So in the concept is that if you're able to target that particular receptor, perhaps it also have overall effect on other interplay of the quorum sensing receptor.
And do you think that when it's actually interacting with that other molecule, that might affect how the drug is able to interact with your regulator? I think that's what Tor's question is asking.
Yeah, if like the MVFR work in the downstream of the last R, and also working upstream of another receptor. There are three main receptors in that particular QSR. So if we're able to like inhibit that particular MVFR, we break the communication with other two receptors in that circle, and overall it will have effect on the inhibiting property of that particular drugs.
Cool, thanks. I was wondering, have you decided how you're going to test your compound? Are you gonna do some testing in the lab to see if it's actually has high efficacy?
Yeah, because we are going to do like biological acid test on it. So we're considering synthesizing them in the lab and checking them out in the lab and see if our computational results complement some of the biological results we are going to get from the lab.
Yeah, cool, fantastic. I hope it goes well. Perhaps we'll move on to the next speaker in the interest of time.
Thank
The next speaker is, thank you so much for that, by the way, Tope. That was a fantastic talk. Our next speaker is Sophie Law KK, and she'll be talking on exploring phage resistance trade-offs to counteract the evolution of antimicrobial resistance in Escherichia coli. Take it away.
Can everyone see my screen? And okay, good morning, everyone. So today I'd like to share with you my research that involves a battle happening at a microscopic level. Everyone, let's imagine this. On one side, we have bacteria, but they are never alone, because on the other side, we have bacteriophages, which are viruses that specifically infect bacteria. Phages attach to the bacteria. They inject their genome and use the host machinery to reproduce. Bacteria will then lyse, releasing new phages. And this natural prey and predator relationship between bacteria and phage has emerged as a potential approach to address antimicrobial resistance, which is the main discussion topic for today's symposium. As we all know, antibiotics are the drugs that can be used to target bacteria, and overusing them will lead to the evolution of resistance, where the drugs just stop responding against the resistant strains. However, using phages to address AMR is quite challenging as well, because similar to antibiotics, bacteria can evolve phage resistance, making them less effective and eventually lose their potential values. And this problem eventually brings us to the research question that I'm tackling today. How can we prevent or slow down the evolution of phage resistance? To answer this, I started with wastewater. Think of it as a house that is full with different types of phages. And from here, I introduced E. coli as my fishing bait into the wastewater and isolated 2 new phages that can target E. coli, and we named them BLS2 and BLS5. Then we went to test how well can these phages infect E. coli. We found that initially, the phages can suppress the growth of the bacteria, but as time passed, the bacteria began to regrow. And why is this happening? E. coli under phage stress can rapidly evolve phage resistance, allowing them to survive and multiply. But to exactly figure out how they are doing so, we isolated and sequenced 12 phage-resistant bacteria, 6 for each phage. And we found that most of the phage-resistant bacteria from both phages mutates the lipopolysaccharide genes to prevent the phage from binding. And this tells us that the phages relies or use the lipopolysaccharides to cause infection. But what exactly is lipopolysaccharides? LPS are molecules found on the outer membrane. Think of it as a coat or a barrier that protects the bacteria from harmful chemicals like antibiotics. We also found that certain phage-resistant bacteria, about 20% of them, mutate other genes instead. And these mutants retain the wild-type complete LPS structures. Another 20% of the phage-resistant bacteria were expected to have partial LPS loss because the affected genes will only allow the bacteria to have some disruption in the LPS, but it wasn't completely lost. And majority of them are expected to have severe LPS loss because the affected genes maybe will allow the bacteria to make tiny portion of the LPS, just enough to ensure their survival, but may not have any protective effect. Indeed, we found that the wild-type E. coli, as well as the phage-resistant bacteria that has complete LPS, were naturally resistant to erythromycin. This antibiotic is not commonly used against gram-negative bacteria due to the presence of LPS. However, for the remaining phage-resistant bacteria that either have partial or severe LPS loss, they became more sensitive to erythromycin. Their resistance actually dropped by 4 to 128-fold compared to the wild-type bacteria. Given the loss of LPS will make the bacteria more susceptible to erythromycin, this leads us to wonder, what if we combine phage and erythromycin? By right, the presence of erythromycin will kill off the LPS mutants that evolved, which means, can this combination slow down the evolution of phage resistance? The white colonies on this agar represents the phage-resistant bacteria. We notice as we increase the concentration of erythromycin, the number of these white colonies reduce. and the reduction was more drastic at slightly higher concentrations of erythromycin, which means erythromycin can really help to slow down the evolution of phage resistance. So far, we know that damages to LPS will resensitize E. coli to erythromycin, but actually this susceptibility is linked to how much LPS they have lost. Those with severe LPS loss were very susceptible to the antibiotic compared to those with partial LPS loss. Mutants or bacteria that still have complete LPS were resistant to erythromycin. So different phage-resistant bacteria have different susceptibility, and this leads us to wonder, can we direct the evolution of the phage to drive the evolution of antibiotic sensitivity, given that phages can evolve over time? Which means when the bacteria evolve phage resistance, can we somehow push it to only have severe LPS loss? Because these changes is most susceptible to erythromycin. To test this, we train the phages using 3 different E. colis. First, we train them using E. coli that has a more complex LPS structure. In the second condition, we train them using E. coli that have complete wild-type LPS but resistant to the phage. And for the last condition, we use the phage-resistant bacteria that has partial LPS structures. First, we introduce the untrained phages to a culture that contains the wild-type E. coli and the new E. coli strains. And after overnight incubation, we purify the phage and transfer them to a fresh culture. By doing so, we only allow the phages to evolve, because bacteria were discarded and replaced at each cycle. And this process was repeated until the phages is adapted to infect the new E. coli strains. But to truly figure out whether this training process can really drive the evolution of the bacteria towards the extreme LPS loss, then we let the bacteria to evolve phage resistance. The figures here compare the ability of E. coli to evolve resistance to both the untrained as well as the trained phages. And from here, we observe that certain phage training process reduce the ability of E. coli to evolve resistance. And the best result seems from training the phages using E. coli with partial LPS structure, because there were fewer phage-resistant colonies when we compare to the untrained phages. Then we sequence a total of 48 phage-resistant bacteria to check whether these mutants really have severe LPS loss. Earlier on, for the untrained phages, we noticed roughly 50 to 60% of the phage-resistant bacteria have severe LPS loss. And this proportion increased to about 85 to 100% once we train the phages using E. coli with different LPS profiles. Again, this type of changes is most susceptible to erythromycin. Given that the evolution of AMR is something that we can't really avoid, but what if we could use the power of evolutions against itself? Through the studies, we have demonstrated how phages can drive the phage-resistant bacteria to regain antibiotic sensitivity, and how we can use antibiotic to slow down phage resistance. And these demonstrated the trade-off between phage resistance and antibiotic resistance to reduce the impact of AMR. And in real life, where we are running low of effective antibiotics, phages may be promising. But again, bacteria can evolve phage resistance. If it happens with trade-off like increased antibiotic sensitivity, then we can use existing antibiotics to target the phage-resistant mutant. But that's not always the case, because bacteria can also evolve without trade-off. They end up being resistant to both phage and antibiotic. And to our knowledge, this is the first study to demonstrate how directed phage training can be used, not only to overcome phage resistance, even when bacteria evolve resistance, we can drive towards a specific trade-off, like resensitizing them to antibiotics. And I'd like to take this opportunity to thank my supervisors, McDonald's Lab, BARS Lab, and Lingos Lab, for providing samples, bacteria, and advisors in these studies. And thank you everyone for listening.
That was a really fantastic talk, Sophie. Thanks very much for that. We already have a question from Reese. He says, great talk, Sophie. Do your phages infect non-lab adapted strains of E. coli, such as pathogenic strains, i.e. UPEC, EPEC, or strains that produce an O antigen?
We tried infecting it in UPEC, but it doesn't work very well. It's too specific towards BL21. You won't test it using other lab strains of E. coli. It is still not infecting. It's quite specific to the E. coli that I'm using currently.
Jamia has a follow-up question. Fantastic talk, Sophie. Do you know if other gram-negative pathogens behave like this, or is the loss of LPS an E. coli specific mechanism?
I think in general, if the bacteria have some loss in their LPS, it will somehow affect their susceptibility because certain antibiotics are like reliant on the LPS to infect the bacteria. Yeah.
I have a question. Do you have a sense of what changes took place in the phage to increase its capacity to cause the severe loss of LPS?
I think to answer that, we will first need to know like which phage gene is responsible for, let's say, attaching to the LPS. And from there, we can study the gene sequence and to see like how we can improve or modify it. Yeah.
So you haven't sequenced it yet. Are you planning to? Is that
We haven't sequenced the trained phages, but we have only the ancestral phage.
yeah.
But we'll consider this. Yeah. Thank you.
In the host as well, when you apply the phage, the bacteria will be present and they'll be evolving alongside the phage too. Do you think that will be an issue?
Because in my training model, I will introduce like the original bacteria because if I only put the phage with the new host, it seems like it's a bit hard for them to adapt. Like,
Yeah, and
no.
that's what will be happening in the host, even when you apply the trained phage. So how do you think that will affect the trained phage?
As in, you mean like how will the phage affect the original host or the new?
No, I mean, sorry, let me
Sorry.
I'm just asking, so in the way you train the phage was really
Mhm.
cool with exposing it only to the original host
It's original and the new host together.
and the new host.
Yeah.
Okay. Okay, cool, makes sense.
Okay.
We have two more questions, one from Saham. He says, great talk. Have you tested other classes of antibiotics aside from macrolides or perhaps other macrolides like azithromycin?
So far, we only tested erythromycin because this antibiotic is not effective against most of the gram-negative bacteria due to LPS, and then we think that this might be the most suitable antibiotic to test. But in the future, we might also test like on different classes of antibiotics.
Yep, that makes sense. And one last question, I think. Phage therapy is an innovative approach to tackling resistance in bacteria, but phages can transfer resistance genes through transduction. How do we select phages for therapy?
In order to select phage for therapy, I think first it has to be lytic, but not that suitable if, let's say, it is like temperate, like it can incorporate their genes into the bacteria. So I think through sequencing, we can determine and also do some testing whether they are lytic, solely lytic, or it could be a temperate phage. Yeah.
Yeah, cool. Fantastic. That was really great, Sophie. Thanks very much for that.
Thank you.
I think that brings us to the end of our session.